Good afternoon, everyone. Welcome to the Wednesday afternoon lecture, today to be delivered by Carol Barnes. When I was 12 or 13 years old, I spent a lot of time in the theater because my father was a drama professor and a director, and my mother was a playwright. And in those days, I could look at a script, read it 2 or 3 times, and pretty well be ready to go on the stage and get my lines right. More recently, I've discovered it's not so easy to do that. A lot more study is required, a lot more rehearsal, a lot more going through the lines with somebody prompting you. So what's that all about? Well, I guess that's partly what we're going to hear about from this particular notable expert, Carol Barnes, notorious, I suppose, as well, who's going to talk to us about memory and the aging brain. And of course, there are many bad jokes about that, and we won't go into those because I would forget the punchline. So let me say a word about Carol. She got her undergraduate degree at University of California, Riverside, BA in psychology, went on to obtain a PhD in psychology at Carleton University, and then managed in a space of just 5 years to hit 3 different countries in her postdoctoral training, including Halifax, Oslo, and London. Following that, she joined the faculty at University of Colorado Boulder, where she was promoted fairly quickly through the ranks to full professor, and then left to join the University of Arizona, where she currently is a full professor with a number of titles. She's the Regents Professor in the Departments of Psychology, Neurology, and Bio 5, and the Director of the Evelyn F. McKnight Brain Institute, as well as Director of the ARL Division of Neural Systems, Memory, and Aging. A lot of directing you must have to do there. I feel for you. And she also has been a very significant leader on the national and international stage, most notably as recent past president of the Society for Neuroscience. As far as NIH is concerned, she has served nobly here by 2 different stints on the National Advisory Council for Aging and on the Board of Scientific Counselors for NIMH. So you're in for a treat as she is going to walk us through a whole variety of different perspectives on information processing and memory that occur during the course of aging, bringing into that molecular biology, electrophysiology, and behavior. So please join me in welcoming Dr. Carol Barnes.
Thanks so much for that. Thanks so much for the wonderful introduction. And it's been really fun to hear you, talk to you today. I had a great student lunch this afternoon. Lots of exciting new things happening with the students and postdocs here. And it's a very vibrant place that I really enjoy coming back to. Okay, the title of my talk and the focus of my talk today, of course, will be on how circuits important for memory and the behavioral expression of experience are altered across the lifespan. Now, the goal here is to understand what happens in the absence of dementing illness as a backdrop for defining disease processes that can be superimposed on what I'll call normative aging processes. Now, the collaborator in the experiments that I'll talk about today, as well as the points that I want to make are here. First, I want to start with examples of the kinds of hippocampal-dependent age-related memory deficits that occur across the lifespan that can be observed not only in old rats and mice, but also in old humans and monkeys. And I'll also review electrophysiological changes in hippocampal responsiveness, connectivity, and synaptic plasticity in old rats. And after that, I'm going to describe an experiment using a method that monitors behaviorally driven gene expression in circuits across the brain that has single-cell resolution. And with this technique, we've begun to assess the functional activity patterns across, changes in functional activity patterns across the lifespan, along with transcriptional alterations that might be mechanisms that underlie these kinds of changes. Then I'll give 2 examples of Ensemble recordings and how the network dynamics of hippocampal cells are altered across age. And because the ultimate goal of my research program over the past several decades has been to help reveal causes of memory decline that are generalizable across all mammals. Over the past number of years, I've begun to study the non-human primate, and so I'll be showing you data from these experiments that are still in progress in young and old monkeys, and I'll tell you about a few small successes that we've had in this regard. The imaging and behavior experiments are the stories that are most fully developed, and so I'll be focusing on those here today. Okay, let's start with memory and normal aging. Well, the hippocampal, the brain structure of the hippocampus and the hippocampal contribution to spatial memory processes is across vertebrate species is widely appreciated. And the type of memory that involves navigation can be tested under similar conditions across species, which makes it particularly useful because it does not require language, and therefore it provides the ability to study memory that's dependent on this brain structure in some detail. Now, what you need to know is that older animals in all species examined, from rats and mice and dogs to monkeys and humans, can show impairments in this type of memory that requires wayfinding. For example, spatial impairments can be observed in this circular platform task that you see here, and we've shown this in old rats, and Bach et al. Have shown it in old mice, and also spatial memory impairments can be observed by allowing animals to perform in the Morris swim task. Rusty Gage was the first one to show this kind of deficit in older animals. And the object of both of these tasks is to remember where escape is possible, either into a dark escape box underneath this platform surface or onto a platform that's hidden underneath the water. To do this, animals use the distal visual cues in the room to guide their behavior and accurate performance. And old rats and old mice show impairments in both of these tasks, indicating changes in spatial memory with age. Now, Peter Rapp, who's now here in the NIA intramural program, also has data that are consistent with the poor use of spatial strategies in old rhesus monkeys, using an 8-choice foraging task. So there's a tether that holds the monkey, and around the perimeter of the platform, the monkey is able to obtain a favored food, and his job is to figure out where he's already been to get the food and then not go back to that place and accurately retrieve the food from around the entire platform. And delays could be imposed between the choices that these older monkeys would make and younger monkeys. So you could tether the animal to the center of the platform. And the observation was that older monkeys became increasingly impaired when the memory demands of the tasks were increased by increasing these delays, again suggesting memory impairments that occur in these old macaques. For humans, I could give you a number of examples of humans, but I'll give you one from that Mary Newman and Al Kazniak experiment that they conducted, in which they used a dry version, what you call a dry version of that Morris water task. They placed a pole into this tent, which was about 20 feet across, and they had people go in and look at the configuration of cues, go out and have to replace the pole. And the older adults were not as accurate as the younger adults in accurately locating this pole, indicating again differences in spatial knowledge between age groups. So the point I want you to take home from all this is that in all the species tested, there are changes in memory performance on hippocampal dependent tasks, that is, animals with damage to the hippocampus cannot perform these tasks. And this suggests that the hippocampus may then be a structure vulnerable to the normative aging process. So a number of factors may contribute to these observed memory deficits, and so we've been interested in determining what these factors are, of course. Well, the good news for those of us who are absolutely determined to age normally and successfully, is that there's widespread preservation of cellular function in the aged brain. You see here sections through the rat hippocampus, so these are intercellular dye-filled CA1 pyramidal cells of the hippocampus. These are CA3 pyramidal cells, and these are granule cells of the dentate gyrus, which I'll be talking about in a moment. Now, for cell numbers, when stereological cell counts are made post-mortem, there's no change in principal cell numbers with normal aging. What do I mean by principal cells? These pyramidal cells and granule cells. No change in numbers, not in humans, not in monkeys, not in dogs, not in mice, not in rats, where these methods have been applied. And Peter Rapp and his colleagues have also shown a preservation of cell numbers in different temporal lobe regions, like the entorhinal cortex, which sends massive projections to the hippocampus itself. So, no cell loss. Now, for electrophysiological properties of aged hippocampal cells in live tissue in vitro, most biophysical properties of old hippocampal cells are quite intact. Old cells are healthy in these slices, and their membrane properties are very similar across not only from data from my lab, from many labs. And so indeed, hippocampal cell preservation is the norm. Now, I don't want you to leave here thinking that there's nothing that goes wrong with the hippocampus. While hippocampal preservation during aging is more the rule, there are both anatomical and electrophysiological changes that do occur, and I'm going to mention some of these changes here. I'm going to go through for you changes that occur in two cell types, the granule cells and the CA1 pyramidal cells. Starting with the granule cells, Yuri Geinesman and his colleagues first showed, using serial EM approaches, that there are fewer synaptic contacts from the entorhinal cortex, the temporal lobe projection, onto these granule cells in old animals. And electrophysiology from my lab has shown that there appears to be axon collateral pruning, and so with these axon collaterals that are lost go synapses. But the synapses that remain in older animals are actually more powerful. They are stronger, so they cause more depolarization in the postsynaptic cell, as though a compensatory process was occurring at the synaptic contact. For CA1 pyramidal cells, on the other hand, Geinesman again did these studies. They showed that there are similar numbers of anatomical synapses from the CA3 Schaffer collateral projection onto these CA1 pyramidal cells. But interestingly, a subset of these cells shows a subset of the synapses shows a decrease in the postsynaptic density size in the older animals. Now, the physiology as well shows no change in numbers of axons, but rather a substantial loss of synaptic contacts along a given branch of axon in the older animals. And there is no change in synaptic strength, so what this suggests is that perhaps these synapses with the reduced postsynaptic density size are silent. This is an interesting finding because the synapses are still there, the cells are still there. This might be a really good therapeutic target. In CA1, there's no compensatory change in this region. So the data that I've just reviewed for you here serves to illustrate the fact that the changes that do occur in the aging brain are exquisitely specific, in fact. They're not widespread in general, but they happen differently in different regions. What about plasticity mechanisms with age? Okay, with respect to plasticity, both long-term potentiation, the increasing of synaptic strength, and long-term depression, the decreasing of synaptic strength, change in hippocampal circuits with aging. Now, the early observations were that LTP at the peripheral path granule cell synapse in older animals decays faster in old rats. So if this is a diagram of synaptic modification that can be induced with this patterned electrical activity, you get stronger synaptic strength. But if you record from these animals over many weeks, you see that the decay time constant for LTP is about 40 days in these young animals, about 20 days in the older animals. And this LTP decay rate, importantly, is correlated with how accurate the animals were on the circular platform task, both in the young and older rats. So spatial memory is good. You'll have, if in either age group, you'll have the strongest, most durable LTP. While LTP can be induced in old rats powerfully, as you see here, that it can be with very strong parameters. In fact, if you use weaker stimulation parameters, it can be more difficult to induce. And a number of demonstrations from Tom Foster's lab, a number of experiments have shown that both LTD, long-term depression, and LTP reversal, which reduces synaptic strength again, is increased in older animals. So you see here synaptic strength in the baseline, stimulation that induces LTD, and the older animals having stronger LTD overall. Now, these data suggest that the balance of increasing and decreasing synaptic weights could be altered during aging. The interpretation of this, of course, still is up for grabs, whether it's an adaptive response to a lifetime of storage of information, because how do we store information? We store information as a distribution of synaptic weights in the nervous system. Well, it could be this, or it could be a breakdown in set point for overall synaptic strength, and these things are still being debated. Okay. Now, before going on to ensemble recording methods for age comparisons of functional circuits, I want to mention, while electrophysiology, that's what I am. I'm an electrophysiologist, and it's near and dear to my heart. And actually, the temporal resolution of those methods are really unsurpassed. The number of cells that can be sampled at once remains a limitation. So even with our improved methods over the years, where we can record really quite a few cells simultaneously, still it's a rather small number. And so because neural encoding is thought to involve variations in which neurons are activated during a given event or which circuits are engaged, we've been searching for an additional method that can distinguish multiple experiences in the same animals over wide regions of the brain at cellular resolution. And after the immediate early gene ARC was described by Paul Worley and others, John Guzowski took up this challenge directly when he was in he was a postdoc with Paul and I. And John was able to demonstrate something very striking and interesting, and that is that that ARC showed a cell-specific pattern of expression following behavioral experience that had a direct correspondence to electrophysiological activity that is known to occur in the hippocampus and a number of other brain regions. That he demonstrated that the cellular compartmentalization of ARC gene expression varies as a function of time from the event that activated it. So the location of ARC in the cell then, whether in the nucleus or in the cytoplasm, could give you a temporal monitor of the activity of neurons, allowing identification of which neurons participated in each of two distinct behavioral epochs in the same animal. And we've called this method catfish for short, because this is a horrible mouthful to spit out all at once. So what we do is to use high-resolution confocal microscopy, and you see the nucleus is blue here, and the red gene product can be observed either in the nucleus as these transcriptional foci here, or, and this occurs from 2 to 15 minutes after behavior, or outside, around, surrounding the nucleus in the cytoplasm from 15 minutes to 60 minutes later. Or you can see it in both compartments if you have behavior, a 20-minute interval, and the same behavior next. So using these methods then, Marcia Penner, as part of her dissertation work, used ARC to assess the extent to which hippocampal circuits engaged in spatial behaviors are altered by the aging process. And actually, she had many, many conditions in her thesis experiments. I'm just going to focus on one for illustrative purposes. She's exposed young and old animals to a bout of exploration for 5 minutes, so they ran around a square chamber, foraging, and we expected, because of the temporal order that the temporal factors involved in arc induction, that this 5-minute exposure, the arc would then be in the nucleus in these animals. And one hemisphere was taken for catfish analysis. The other hemisphere of the same animal was taken, it was dissected out to quantify ARC and CA1 by real-time RT-PCR. And to make a long story short, the total numbers of cells that are activated and showed ARC expression by the catfish method was the same in the young and the older animals. And the majority of ARC-expressing cells showed ARC expression in the nucleus, as would be predicted by these methods and the temporal characteristics of these methods. So for the PCR, there was the fold increase in ARC mRNA following behavior was also similar across age, but the relative amount of ARC RNA was less in old than in young rats. And this suggested to Marcia that some or all of aged CA1 pyramidal cells may transcribe less ARC. So to determine whether we could detect differences in intensities of these transcriptional foci, actually it was Paul Worley's idea, well, maybe we could just measure the intensity of these foci and in young and old rats, and we did. So the mean level, intensity level was reduced in aged rats, and as you can see, the histograms of the intensity are shifted. The distribution is actually shifted left to lower intensities, suggesting to Marcia that perhaps then some or all of aged CA1 pyramidal cells express less ARC. Now, what could be the mechanism for this difference in transcription? And one mechanism that can regulate transcription is, of course, DNA methylation. So we initiated a collaboration with David Sweat's lab to investigate whether the attenuation of ARC that we see in aged neurons might be modulated by this through some transcriptional repression. So to do this, we, with David's help, did direct bisulfite sequencing PCR to examine changes at 2 loci of the ARC gene. The focus was on 2 regions in the ARC promoter region, so 2 CPJ islands, either the promoter region or this intergenic region. And interestingly, the first thing I want you to notice about these data is that even with this mild behavioral treatment, that is animals foraging behaviorally, just doing general navigation, there's a decrease in methylation in the promoter region of ARC and the CA1 region. So there's a decrease in both young and old rats, consistent then during behavior with a permissive environment for ARC expression. So just 5 minutes of explorative behavior can rapidly and dynamically change the methylation state of this ARC gene in old rats' pyramidal cells. When Marcia looked downstream of this promoter region in this intergenic region, again, in this case, you should also notice that both young and old rats showed dynamic methylation changes in methylation state as a result of behavior. But in this case, notice that for the young rats, let's focus on this first, young rats show an increase of methylation after behavior. So you might ask, well, why would increased methylation after behavior have a positive effect on transcription in this specific region? Well, we think that part of the answer might lie in recent work by Chara et al., who have observed that DNA methylation, of course, is not always associated with transcriptional repression, but can indicate active gene transcription events in brain, something that's long been appreciated, of course, by cancer biologists. So our hypothesis now is that optimal ARC transcription may require 2 events in CA1, demethylation at this promoter region, methylation at this intragenic region of the ARC gene. Now, consistent with what we saw in the promoter region, notice that aged rats also show a higher baseline level of methylation than the adult rats in the rest state. But following behavior, when young animals show an increase, the older animals show a decrease. So if our interpretation is correct, then the combination of these changes would lead to more transcription in the younger animals than in the older animals. What might it mean that there's less ARC available in older rats? Well, there's a number of things that we might suspect could be affected. First, we've shown that knockdown of ARC in behaving animals reduces the durability of late phase LTP, affects memory consolidation, and also ARC knockout mice show impaired maintenance of LTP and defective spatial learning and fear conditioning. And additionally, Worley, Hugener, and their colleagues have described a role for ARC protein in AMPA-like glutamate receptor trafficking and mGluR-dependent LTD, and also a role in homeostatic scaling processes. Now, while the precise relationship between ARC's role in LTD and the maintenance of LTP remains to be determined, if aged cells in the hippocampal networks transcribe less ARC, this could alter plasticity mechanisms that could significantly change hippocampal circuit dynamics. And circuit dynamics is where I'm headed now to talk about electrophysiological recordings using Ensemble recording methods. So how do we record from our cells in individual hippocampal cells from a freely behaving rat? Well, we start with this manipulator device called a hyperdrive. It lowers 12 different tetrodes, these four-pronged recording probes. And with this method, we can isolate a number of cells at once from each tetrode using a number of cell separation techniques and offline clustering analyses that allow us to triangulate in and identify single cells. Now, with this method, we can examine up to 100 cells or more at once in the freely behaving animal, but even on just medium days, we can record from 50 or more, which is quite an improvement over the 2 or 3 that was possible maybe even only a decade ago. So what I'm going to show you now is a simple illustration of the observation that single cells in the hippocampus, so that here's one of these CA1 pyramidal cells. We're listening to one of these pyramidal cells. The rat is running around this large arena. He's tethered to the recording system. He's got light-emitting diodes here that you can see. So when John O'Keefe first noticed this property of hippocampal cells in the late 1960s, you can see why he called these cells place cells. And here you see the place field where the cell fires. The area over which the cell fires is called the place field in yellow dots. And presumably then the firing pattern of hippocampal cells will tessellate across the entire environment and provide what O'Keefe and Nadel have called a cognitive map of a particular space. Now, when I refer to a hippocampal map, I simply mean the distribution of place fields in a given space. Okay, so I'm going to give you one example of an age comparison looking at these place fields. And when hippocampal cells in young rats are recorded, so this is a triangular track, and the rat is running around like a good rat and getting chocolate milk in the centers of these arms, so he kind of looks over to see if there's any other chocolate milk, and he runs around in the first session. And then in the second session, he's been taken out and put back, and what you see is a distribution of some place fields that we're being recording from. So there's a yellow cell here, a red cell here, and so forth. So after the second session, what you should notice is when the rat goes back in and runs around the maze, the same map is retrieved. The yellow cell still is firing here, the red cell is still firing here. And if you look at old animals, if you take an old animal, and if you just focus on the upper graphs again still, about 2/3 of the time you put an animal in, record from an old animal in this session, and then take him out, put him back in, you'll see that the old animal also retrieves the same map, very much like this. But on about 1/3 of the time, for any given old rat, when you record from him on the first session, you see this blue cell is firing here. You take him out, record again on this session. It's as though the animal had retrieved, or the hippocampus has retrieved, an entirely different map. So the blue cell now is firing here, and this orange cell didn't fire at all on the first session, as though the old rat had retrieved the wrong map. Now, because of time, I'm not going to show you what happens in CA3 pyramidal cells. This is for CA1 pyramidal cells. But I'll just tell you that under conditions when you have two environments, but the environments are completely different, and young rats remap, sometimes old rats reuse the same CA3 map. Clearly a different kind of retrieval error than we see here in CA1. These results taken together suggest that dynamic processes of pattern separation, pattern completion in the hippocampus are altered in these networks, and that the old rats sometimes, in some cases, retrieve a different map when they shouldn't, and in other cases for CA3, inappropriately retrieve a preformed map that they should not reuse. CA3 data is data from Ian Wilson and Howard Eichenbaum's lab. So if you can picture this, if you've got a map of Washington, D.C., you're trying to find your way around Bethesda, you're going to not have an easy time of navigating. So at the very least, this indicates that integrating episodes into the contexts in which they're experienced will be more difficult for old animals, and may be partly responsible for why older organisms get lost. The second example of how age may impact hippocampal circuits involved in spatial cognition comes from the comparison of what's been called experience-dependent place field expansion plasticity in young and old rats. Now, when a young rat repeatedly traverses a route, the pattern of place cell discharge changes as a consequence. And unfortunately, Mayank Mehta gave it this hugely long name. It's a mouthful, but it's place field expansion plasticity. And so if you look at the rat moving this direction on the maze, the first time he goes through the cell that you're recording from place field, it might look like this. But after repeated exposure and repeated routes on this maze, the field will expand in the direction opposite to the direction of the rat's movement. And this was consistent with theories going as far back as Hebb's phase sequences, which are cell assemblies that are connected together that could serve associations and encode sequences or episodes. So Arne Ekström showed that this place field expansion plasticity, which happens normally, there's no patterned activation here, but this is apparently a normal form or a behaviorally induced form of LTP. Arne showed that this is NMDA receptor dependent, as is artificially induced LTP. And when Jaemin Shen, for a dissertation, looked at this plasticity mechanism in old and young rats. So these are the numbers of laps across numbers of laps and the amount of place field size change that you get. You see that there's a robust plasticity in the younger animals and much less robust plasticity in the older rats. So she suggested then that this is consistent with the likelihood of a failure of effective naturally occurring LTP-like mechanisms in old rats, and importantly predicts weaker memory for sequences. Now, one major change in aged CA1 cells that I didn't yet tell you about is a disruption in calcium regulation. And prominent contributors to the literature in this field are Phil Landfield and Tom Foster. But there are more L-type calcium channels in old CA1 pyramidal cells. And Sarah Burke and others in my lab realized that, if calcium influx through the NMDA receptor could be modified, perhaps it would be possible to return the plasticity characteristics of these cells towards a dynamic range normally shown in young animals. And this brought to mind the agent memantine, which is a low-affinity, non-competitive NMDA receptor antagonist, which blocks the open pore of the NMDA channel. And it's currently used as Namenda, I think, to improve cognitive function in some stages of Alzheimer's disease. So these improvements, you may know, in cognition in humans and animal experiments have been explained by the idea that memantine may act to increase signal-to-noise ratio under conditions in which normal synaptic transmission has been disrupted. And for old rats, this would mean calcium dysregulation. So Sarah decided to try memantine to see whether she could change the place field expansion plasticity, and what she was able to do is see that in the saline rats, compared to saline, old rats given memantine did show improved plasticity. At least the plasticity was augmented, suggesting that at least acute administration of a clinically relevant dose of memantine may partially reinstate this naturally occurring LTP-like plasticity mechanisms and may aid in normalizing the circuit dynamics within the hippocampus. I'm going to change gears a little bit now to talk about my non-human primate work. Most of what's known about the detailed physiology of hippocampal neurons in awake behaving mammals, especially aged mammals, has come from studies conducted in rats and only more recently aged non-human primates. So for the last part of my talk, I want to show you some data from an ongoing experiment or study we've been conducting at the California National Primate Research Center at Davis using 6 young and 6 old rhesus macaques. You might wonder, what's an old rat? What's an old primate? Well, an old rat is 24 months of age for a Fisher 344 rat is equivalent to about 70 years of human years. And for primates, you have to multiply by 3. So a 25-year-old monkey macaque would be equivalent to about a 75-year-old human. Okay. So what we've been doing with the 6 young and 6 old rhesus monkeys is collecting behavioral imaging, electrophysiological data, as well as tissue for molecular and biochemical analysis from these animals. And we always send all the tissue of the other body parts at histology to NIA's data bank for banking for muscle and heart and other things. So what we've begun to do is to, in terms of the physiology, is to use Bruce McNaughton's hyperdrive that we've modified to use for non-human primates, for monkeys. And the hyperdrive and these tetrode recording probes have been extremely successful. We have obtained, with one tetrode probe, we've obtained up to 20, and actually our new record is 25 cells at once from one tetrode. And this is because of the high packing density of the hippocampus. There's many cells from which your tetrode can sample. These data are from Bill Skaggs's manuscript. We also record hippocampal cells that are complex spike cells in the primate, and it's these complex spike cells that you heard firing as place cells in the rat. So they show the same cell type in the hippocampal CA1 region as well as the rat, and the mean firing rates over behavioral sessions in cells that we've recorded are well below 1 hertz. Again, very, very similar to that we've recorded in the rat. Even when recording from our 32-year-old macaque, this would be, she was equivalent to 96 years, human years. So the firing rate is stable and low, like you see here, even in the older monkeys. Several young and old monkeys, we've also established that there is a strong relationship between the hippocampal EEG amplitude when the behavior is when the animal is off task and drowsy, you get these large hippocampal sharp waves. When the animal is in an active attentive state, you do not get these, and we were able to show in a depth profile through the hippocampus that's reconstructed after histology these sharp waves events occurring that peaks in the hippocampal fissure and reverses in CA1. This is exciting to a physiologist because this is exactly the same kind of waveforms and responses that we see in our rats as well. So the similarity of these sharp waves, the hippocampal cell types that we see, and firing rates at least encourages us that what we've perhaps what we've learned in the aging rat may be informative for primates, but now we'll be able to determine this directly as we finish our studies and compare these kinds of network characteristics across age in the non-human primate. I know there are a number of you in this room that work with non-human primates, and this work is really hard. It proceeds at the pace that it proceeds, which is slowly. And so I'm not done with the full age comparison, and I won't be for a number of more years, but it will be a classic. It will be a classic in the field because nobody has looked at these kind of things in the aged monkey. So the experiments that I'm going to focus on today involve behavior and imaging methods, and it really continues on the theme that I've been trying to carry throughout this talk, that cognitive dysfunction can occur in the absence of large changes in neuron number. I think all of you have heard that you lose at least 10,000 neurons a day and so forth. And if you want to ask me about it at the break, I'll be glad to tell you where that all came from and why it's wrong. But again, in the same population of aged and young animals out at the Primate Center, David Amaral helped me, along with Mike Bonacore, to do first MRI scannings in these young and old monkeys. Julia Shami, when she was in Peter Rapp's lab, did standard volumetric analysis so that the hippocampal volume could be measured in these animals and compared over the age range of 9 to 29 years of age. And in fact, what we found was that the volume, hippocampal volume, differed by less than 6%. So not surprisingly, then, there was no relationship between hippocampal volume and the memory deficits that we observe in these monkeys. This is an important observation, I think, because of all animals, it's only humans that get Alzheimer's disease spontaneously. And so we're looking at a living animal who by definition is Alzheimer's disease free. And the thing I'm bringing up is that with human imaging experiments, there's always some uncertainty about whether the person in the experiment who you're examining is disease free at the time of testing, will remain disease free, 10 or 15 years hence. So it's at least more difficult in human studies to be certain that you're testing a population that's aging normally. But at least in these aged monkeys, there's no change in volume in the hippocampal region. Now, these same animals, again, were imaged using a technique that Scott Small has helped us with that relies on measures of cerebral blood volume rather than the blood flow measures that I just showed you. And this CBV measurement reflects basal or resting levels of metabolism. And because higher resolution methods can be used with this technique, we're able to segment out, say, the dentate gyrus, subiculum, and CA1, and the entorhinal cortex separately, and examine whether there was differences in resting activity levels between young and old monkeys across these subregions. So again, these are the same animals that showed no hippocampal volume change, and Scott Small was able to show that across age, there was no correlation between age and the amount of this CBV or metabolic activity in the entorhinal cortex. No change in CA1, no change in the subiculum, another region, the output region of the hippocampus. But there was a significant negative correlation between activity level and age in the dentate gyrus, the region with granule cells, with older monkeys showing lower activity levels. And most importantly, there was also a region-selective significant correlation between how much metabolic activity there was and how well the animal did on a memory task, a recognition memory task. The higher activity levels, the better the memory, suggesting that the dentate gyrus may be particularly vulnerable in normal aging. Now, we know that MRI measurements do not have single-cell resolution and are only an indirect measure of neuron activity. And greatly encouraged by our ability to go back and forth between the kinds of age-related changes that we find in the rodents and those seen in humans and non-human primates, Monica Chawla went ahead and applied this catfish technique that I talked about earlier to examine behaviorally induced ARC expression and the composition of hippocampal networks during aging. So what she did was, remember that animals would run around and forage and explore a box, and she provided them with 2 5-minute bouts of exploratory behavior separated by 20 minutes. And what she found was when she counted the proportion of ARC-positive cells in the hippocampus in the different regions of young, middle-aged, and old rats, you see in CA1 there was no difference in proportions of cells that expressed ARC. Neither was there a difference in the CA3 region. And this is consistent with the 5-minute exploration data that I showed you from CA1 earlier from Marcia Penno's data. However, aged rats did show significantly lower proportion of ARC expression in the dentate gyrus granule cells, so fewer granule cells were active after behavior. So this suggests that for the rats, just as in old monkeys, the dentate gyrus is a hippocampal subregion particularly susceptible to the effects of advanced age. So to take this story just a bit further, with the help of David Sweat's lab again, Marcia Penner found that the methylation state was also dynamically regulated in these granule cells, and that, again, the aged granule cells had a higher methylation state than the younger granule cells, which you see here in the ARC promoter region, suggesting a state in the older animals not as conducive to transcription, which could be one factor contributing to why we see fewer granule cells expressing ARC in relation to behavior. Now, of course, the extent to which these methylation and demethylation processes are involved in regulating learning and memory needs a lot more rigorous examination. But these data and other data suggests that if we want to understand why older organisms older organisms show suboptimal cognition, some show optimal and some show suboptimal cognition, my bet is that alterations in epigenetic mechanisms are going to be front and center among the important contributing factors that may explain some of the individual differences that are observed between animals. Now, these granule cell data that I've just shown you in the rat and the granule cell data and the metabolic data in the aged monkey suggest that the hippocampal circuits that show the most distinct changes during normal aging appear to be centered, will at least involve the dentate gyrus. And this is very different, I want to bring up, than the pattern of networks known to be targeted by Alzheimer's disease. That is, in Alzheimer's disease, it's these CA1 cells that are particularly vulnerable and die earliest. And in normal aging, it appears to be these granule cells that are altered, while actually in Alzheimer's disease, they do not show changes. These granule cells don't show changes until very late in the progression of the disease. And so I think that this favors the idea, at least, that while normative aging and Alzheimer's disease processes can co-occur, they can have an independent neural pattern, a distinct and independent neural pattern. So let me just try to summarize. I hope I'm staying on time. What I've told you today. So although the changes in memory are not devastating in normal aging, they can be observed across a wide range of mammalian species, even those animals that do not spontaneously get Alzheimer's disease. Now, in the hippocampus, while there's little cell loss, there is a change. There are changes in circuit connections. There's changes in plasticity mechanisms within these circuits, including transcriptional regulation, and hippocampal networks show altered properties such as map retrieval errors and deficient place field expansion plasticity. Imaging markers, including deficits in metabolic activity in monkeys and actually in human dentate gyrus, and deficits in gene expression in single cells, granule cells, suggests that there's altered network participation of these granule cells. And finally, it appears that the networks involved in normal aging and those that are involved in Alzheimer's disease are clearly independent and non-overlapping. And I'll stop there. Thanks. Oh, can you put my acknowledgements back up? Sorry. Yeah, thanks. Okay.
So thank you. We have time for some questions. There are microphones in the aisles. Please approach them if you'd like to pose a question to Dr. Barnes. After hearing a lot of different kinds of science on different animals, I'm sure there are things that people could pose. Yes, go ahead.
So I was wondering, what is the sensory input for these place cells? And is there any evidence that during aging, the sensory input is actually not changing?
No, there's every evidence that every sense that we have does change as we age. Hearing, vision, somatosensory, olfaction, and does change. And the hippocampus receives input from association areas across the brain, wide association areas across the brain. And what's interesting is that if you look at these place cells in old rats, the place cells are perfectly formed and fine and comparable to young place cells until you do a manipulation, such as remove them from the room and come back. And so why we think that it's really this map or network stability phenomenon that is altered, and that it's not purely a sensory, it's not a result of sensory changes. But of course, all senses do change as we age.
Richard.
Carrie, you were emphasizing the difference in the cells which are predominantly affected in Alzheimer's, for example, the CA1 cells and the granule cells, with these other models of aging across species. Can you comment in the models intended in mouse by genetic manipulation, for example, to reproduce the lesions of Alzheimer's disease? Has there been study to identify whether one does successfully recapitulate the change in predominant cell change there?
So I have a lot of great colleagues that have these mouse models, and some of them are probably in the room. But amyloid deposition alone doesn't cause cell death, and I think Frank Lafurla was here recently, and actually the combination of the tau and amyloid, or the triple transgenics with presenilin as well, while he, I think in his earlier studies, got some cell loss, I think in the current models there is not cell loss either. So, but the argument for those models, in favor of those models, is that there's cell dysfunction in those regions, and clearly plasticity mechanisms are altered with the increased amyloid, and tau, and behavior is altered too. So while it doesn't lead down to the cascade to cell death, there is altered functional characteristics. Some really interesting new work from Leonard Muki's group at the Neurological Institute, I was just there reviewing his program, is that there may well be, since I'm an electrophysiologist, this makes me really excited, there may well be electrophysiological changes that are induced by amyloid burden. Because in his knock-in, APP knock-in, and his ApoE4 knock-in compared to ApoE3, you see electrographic seizures in these animals. Now, it's not a full tonic-clonic seizure. You have to record to see them, but there are electrical disturbances. And so they're going on to now try to record from Alzheimer's patients just to see if there's these submotor seizures that can be detected. Of course, those people have a strong amyloid burden. So this could be partly one reason why Alzheimer's patients on some days are quite lucid. Maybe they're not having these electrographic activities, and other days they're not so lucid, and maybe they are having these. So you really need to look at the circuit dynamics overall to see whether these models are recapitulating some of the things in the human patient, and there's some really great work going on there to try to do that with their mouse models, and I'm sure in other places.
A question relates to the hippocampal region and the stem cell turnover. Does the hippocampal cell has a higher turnover rate than the other neuronal cells in the brain?
So one of the most remarkable things about granule cells in the hippocampus is that there is neurogenesis there. There's also neurogenesis in the olfactory bulb. Granule cells do grow, and newly born granule cells do mature and enter and act, or participate in neuronal circuits. And the question you might ask is, well, if there are new granule cells born, why isn't there a change in the number of granule cells over age? And my answer to you is that the granule cells are among the most numerous in the brain. And so counting any kind of stereological count, even if you have a 5% error, the change in number might be, cumulative change of those granule cells that survive might be within that 5% range of error. And it's those kinds of numbers that we're talking about. What's also hopeful about granule cells, and that may be a center of change in aging, is that if we could manipulate the survivability of granule cells in older animals, maybe we could have a larger circuit participation of these granule cells, and maybe it would help with cognition.
So in terms of the age effect, is primate the best model for a human? Because there are changes in primate that does not happen in the other way. Like say, the primates do not have menopause. So there are other physiological changes that is important in primates, difference in primates and humans.
Right. In rats too, they have, and they have a different kind of alterations. I'm embarrassed to say in front of this audience, but my rats have all been male that I've tested. My first real experiments that have balanced female and male organisms have been my non-human primates, and so I have female and male primates that I'm studying, but I can't comment on that at this point.
Carol, is it possible to mark or somehow lineage track the newly generated cells in an older animal and ask whether there's any detectable functional difference between the new and the old cells?
Yes, and we're really excited to be able to do that. I mean, we did this experiment with BRDU, so the newly born cells with ARC expression, we know that some of these cells are incorporated in the network by 5 months or so after the injection. So we think that they're very long-lived, and a postdoc of mine is just finishing a study trying to look at the difference between how long and how successful these activity markers go on. But obviously, because we have ARC expression decreases, we're predicting that probably the survivability is different in these older granule cells. And there's some really nice studies going on here labeling. Somebody was talking to me at lunch about neurogenesis and labeling cells at different ages and fluorescing them differently and seeing how they progressed. And they're going to be doing it right here in mice, I think. These studies are really long and hard, so she has to wait 2 years for the definitive result, but I think they're going to be very, very interesting indeed.
ARC is obviously a very interesting molecule to track, and apparently relatively straightforward to do so, at least in your beautiful pictures. But do we really know what ARC does, and how safe is it to generalize from what's been determined about its methylation status to what's going on with other immediate early responders?
Right. And ARC is a very, special immediate early gene. I think that we know. So the reason, my answer is that, we don't know how the AMPA receptor trafficking, that pulling AMPA receptors out of the membrane, and the maintenance phase of LTP and how we can alter that maintenance phase by knocking down ARC are related and in the transgenic. So we don't really understand completely, although we have some hypotheses about the endocytotic mechanisms and its role in homeostatic scaling. But it is a very special gene in terms of this activity correspondence. We've looked very carefully at CFOS, for example, ZIF268 and HOMER1A, and it's really ARC that goes along with the proportions of cells that are active during behavior. So what's very special is that if you have a behavior, a very defined behavior, and you look at how many spikes you get during that defined behavior, and then you look at an animal just at rest in its home cage and look at how many spikes you get during these sharp wave states. It's the same number of spikes in both conditions. But ARC is only expressed when the animal is firing in a discharge pattern as though these place cells were active, so in these burst patterns. So it's the same number of spikes, just a different patterning. And it's really locked to behavior, attentive behavior. So there's something very special about it that doesn't occur necessarily in these other immediate early genes, and that's why we're excited to follow up on it.
Well, great. There will be a reception in the library. You're all invited to come and have informal conversations with our speaker, and let's thank her again for a wonderful presentation.
Thank you. Thank you.